going to take the theme of the session seriously and provide some framing remarks around global infrastructure and innovation. I'll then drill down and give you some unpublished research results that are coming from such collaborative networks that touch upon some of the questions that were raised earlier this morning. So, shared realities. I think these are relevant because it defines why we might be interested in doing anything globally together. I'll offer two for your consideration. We all must partner with biology to make more. And also, making anything with biology, with apologies to Hobbes, is nasty, brutish, and long as a process. So let me give you some examples. This is from the Department of Energy in the United States circa 2007. It looks at our civilization as we attempt to operate it, where we source many of the base materials from fossils, petroleum, natural gas, and so on the left, in order to produce the things on the right, so textiles for carpets and many other things. And there's this dream that we would transition our civilization from a framing or workflow that looks like this to, as they described in 2007, something that might look like this, where on the left you have sustainable renewable feedstocks, basic biological manufacturing that then goes through a biocatalysis or transformative layer or layers to produce the same set of things, but with a different agile, perhaps more responsible footprint. Doing this is a tremendous amount of work. There's huge pressing needs around pulling this off and so on. So let me switch then to the brutish and long. Most projects in biotechnology today, most partnerships with biology to make things require a Hercules. A famous contemporary example is the work coming out of Berkeley, where colleague Jay Kiesling identified a need to produce a chemical, artemisinin, currently sourced oftentimes from the bark of a tree. Instead, could you use yeast and a modified metabolism within yeast to take sugar as a feedstock and produce this chemical, reducing the dose from $1.50 to 10 cents? Answer turns out to be yes. As George Post mentioned this morning, it was about 150 postdoc years of labor. So that's 15 PhD level experts working for a decade to make one chemical by retuning the metabolism of baker's yeast. The money isn't such a big issue, although it matters. It's the fact that the money needs to be identified and mapped to a global need, and then somebody needs to be knowledgeable in the science and engineering of how to do this, and then they need to be a manager capable of organizing a team and sustaining its operation for a decade. And when you start to combine all of those requirements to make one of these projects true, it's that collectively which makes such work quite difficult. And it's amazing to me that just like in pharmaceuticals, if you look at bio-based manufacturing, the number of new products that are coming to market are identifiable in small integer number values. So we're not doing a lot of this. And if you go back to that big workflow where we'd like to transition our entire civilization, we might have enough money to do it, but we don't have a human capacity to organize all the work right now. That leads to, I argue, a shared opportunity, produce platforms and process improvements that make the engineering of biology much easier. Also, contribute key elements of these capacities to the commons, or the public domain, or the commonwealth. Not all of them, but key elements. This has already been proven for people who follow the field of synthetic biology by students. So I submit for consideration a synthetic biology project that came from the United Kingdom a few years ago. This was done by teenagers at Cambridge University over a period of 16 weeks. They were able to engineer strains of E. coli, 7 different strains, to produce 7 different pigments, from things derived from carotenoids on the left to stuff via violacin on the right. How did they do it? They decoupled design from fab, as we heard about. So they were able to get de novo synthesis of the genetic reagents they didn't already have for free from a gene synthesis company. They also got about half of the genetic components they needed to pull off these biosynthetic pathways for free from a pre-existing collection of parts, the so-called registry of parts. And then they got from DuPont an engineered strain of E. coli that overproduces some of the precursors needed to do bright carotenoid biosynthesis. So this is an example where by sharing some resources and by changing the workflow, decoupling design and fab, you can create a success of do-it-together biotechnology. And this allows people to do much more quickly. So I just want to say this out loud again. If the Kiesling project is one biosynthesis process in yeast, 25 megabucks, 150 postdoc years, this represents 7 biosynthesis processes, 1 summer, about a half dozen to 12 teenagers, 25 kilobucks, right? So with new immature tools, you can have dramatic improvements in the pacing and costs of doing stuff. If you don't know about iGEM, this is the community that gives birth to some of these early piloted resources. This is the so-called Genetic Engineering Olympics for college students. that seems to be continuing to grow globally. So I was surprised, for example, that the numbers for iGEM in the 2011 season have gone up by another 20 to 30%. I thought it would saturate like the VLSI electronics courses on which we designed iGEM at around 120 schools. So what's then our next strategic move beyond amateur academic competitions? Well, an obvious thing to say is we should professionalize this research to get better tools and concepts. And the list of the sorts of things you could imagine doing is quite long. I'll show 5 here, but you could probably go all the way to the end of the alphabet and then start over. So you want rapid prototyping of designer genetic material. You want CAD tools for managing complexity in biology. You want expanded directed evolution tools. You want to play with the chemistry so that biology can make things that go well beyond carbon, nitrogen, phosphorus, and sulfur. You want standards that coordinate work across time and place. So how would we think about what we would first do if this is just a quick list of what's a much longer one for all the things we might imagine? So to begin to prioritize that, I want to begin to return to some of the work from this morning and map this to the reality of practice of genetic engineering. This is an illustration of how I'm going to get to it. It's a gap. This gap is about 4 meters. We have a much bigger gap in biotech. So most biotech systems as they exist today are encoded by 20,000 base pairs or less of designer DNA, where human beings are taking responsibility for encoding the information they want, producing the phenotype you want. That's about a dozen components, maybe a little bit more sometimes. Meanwhile, we've seen ongoing advances at synthesis and construction of genome-like fragments of DNA. These synthesized genomes are not designer genomes. They're recapitulations of natural sequences. And the largest project I'm familiar with comes from Mitsubishi, where they've made almost an 8 megabase fragment of DNA about 5 years ago. So if you just take those numbers as points of reference, and you could find different numbers and tweak it, but the point's still the same, you take 8 million divided by 20,000, and this is what I'll call the biointegration gap. It's a factor of 400. If any of us could teach the others how to become 400 times better at putting the molecules of life back together to do useful things, we would become orders of magnitude better at biotechnology and partnering with biology to do things now. How do we bridge this gap? I don't know. But let's think for a moment why we might want to bridge it. And it's going to be fantastical in thinking. So to use literature as a way to motivate your thoughts, if you are given 20,000 characters to play with in language, you can compose things like a presidential address, an editorial in the newspaper, or a research article in a magazine for a scientific audience. So short declarative statements with some support, but not a lot of deep detail. If I give you megabases or mega characters to compose with, you could create plays, a totally different type of work. You could create an American novel. You could even encode a Russian novel, right, which is going to be less than 5 mega characters, including spaces. So the future of biotechnology, if we can bridge this gap, gets us to a world where we have forward engineering at the genome scale, which means that we should imagine to be able to do everything biology can make encoded by entire genomes. It's not just bulk manufacturing of fine and commodity chemicals, it's program pattern formation, macroscopic engineering of structures, and so on. How do we bridge the gap? Well, it's going to have to come back to the questions from the morning and structure our partnerships globally, I suspect. So I'll make an argument that we should be doing one thing next: develop and populate functional composition frameworks sufficient to enable genome-scale engineering in the organisms you care about. So bridge the gap by figuring out how to forward engineer genomes that actually work. To do this, it seems like you need to do at least 3 things. Rules of composition for putting parts together, dictionaries of functional elements. A dictionary is required to have a language that works, and reference standards for coordinating activities across time and place. I'll unpack each of these. Note that as we do this, just as a sidebar, that this work will ship as information. It won't ship as physical material, and that's significant, just like with the gene sequencing and genome sequencing projects. Because it's going to be shipping as information over networks, we can distribute the work globally and share the load in doing this, figuring out how to design and build genomes. All right, so as a way to get into this quickly, I want to give you an example of reliable composition of a language and a language. So this will be from computer programming. Many folks here can probably process this bit of computer code: a command, print, quotes, a text string, hello. And when you hit the carriage return, what comes on the screen or on the paper is hello. If you change the text string to a different word, you get the different word coming. If you change the text string to whatever you want, with very high reliability, you get that text string displayed. So here's an example where you have a composition framework in a language that allows you to do forward engineering of commands of a machine with near 100% reliability. Let's take this example and then try and map it into genetic engineering. What's the genetic engineering print command? There's many. I'll give you a simple one to think about. Let's say I want to express a protein in a bacteria. I'll have a promoter for initiating transcription. I'll have a 5' untranslated region for directing the initiation of protein synthesis. I'll have a start codon, the coding sequence for my gene of interest, and a stop codon. So the start and stop are like the brackets or the quotes. And the promoter 5' UTR are like the print command. Print this protein in the cytoplasm of a cell. And if this composition of functional elements works, I'll get the protein. And if I change the coding sequence to coding sequence number 2, I'll get protein number 2. And if I do it to coding sequence whatever I want, number 3, I get protein number 3. If you look at genetic engineering, there's not a lot of good data on this. The reliability of this composition is, rough terms, I'd guess about 70%, meaning 30% of the time you don't get your protein translated. Never mind what the protein does once it's made, but you just don't get it. Why is this? Well, here's some new data that begins to look at how messy composition of parts is. And I'll start first just with composition and not talk about changing context, but I'll get to that in a minute. So this is a panel where we're combining all of the most frequently used and abused transcriptional and translational control elements in E. coli in all combinations, and then using those elements to print 2 proteins, a green protein or a red protein on the left and right. So this is basically what most people working with E. coli tend to use. And what you can see is if the color's blue or red, it's low or high printing. There's some interesting things that come from this. So for example, this is a translation start signal. Each column is a translation start signal. This one comes from Philadelphia, Pennsylvania. And you can see that when you combine it with a transcriptional signal from Europe, it doesn't work very well. But it works okay with most others. Here is a translation start signal with RFP from Life Technologies. It doesn't work at all with this European promoter, but it works really well with a different promoter coming from a different source. This B0032 is the translation start signal that most iGEM students use. And it has varying activity across the full set of promoters the iGEM students use. So when they go to make something, the chance that they're actually going to get what they expect, they don't even know what to expect. You're going to have a factor of 10 or more difference in the protein level, just for one protein. And then it doesn't align at all when you begin to move it over here. It gets even worse, as was noted this morning, when you take this same panel and you move it into, in this case, a different strain. You run those cells on a feedstock that's relevant to industry as opposed to an academic researcher, and you get a different footprint. All right, so this is the hairball that we're beginning to work with. Now, what's significant about this data is it's some of the first data where you can actually quantify how messy the hairball is. Why is this a hairball? So let's go one more level to the science. This is an example of an mRNA where you have the beginning of the mRNA coming off the promoter, and this is where the variable region, your favorite protein's coding sequence is going to be. And the control element for initiating translation, you can see, is encoded on the mRNA. And what's happening is the ribosome, which includes RNA, will have a base pairing through the 16S ribosomal RNA at this region here. If anything happens to this molecule, as a function of variation in the sequence down here, it could disrupt the interaction between the ribosome and the initiation of translation. So I'll show that by borrowing a figure from some nice work from UCSF. In other words, the RNA is not necessarily this nice linear polymer. It's going to fold up into some structure depending on what it would like to be. And depending on how that structure folds, the place where translation will start could be buried in a secondary structure, and that secondary structure could be sensitive to the sequence that comes after, which is the sequence that encodes the thing you want to make. And that's going to be changing, much like the text string in a print statement. A very nice paper in Nature Biotech came out exploring this, where they used free energy calculations to try and figure out whether or not they could have computer models that would predict what would happen. And I'll just highlight keywords from the text. There's an absence of modularity between a translation start signal and a coding sequence that's undetermined ahead of time, because there could be the potential for strong secondary structure to form as the coding sequence changes, and you can't get that right ahead of time. Taken together, our results suggest that reusing the same well-characterized putative standard biological part is garbage. It's not going to work. Their suggestion is they use a calculator, so every time you want to express a particular protein, you're going to simulate in that context the likely expression level and then tune the bases to get the expression level you want. And just to summarize how good those calculators are today, so you have a sense of this, the error coming from these calculators for translation levels has a 47% chance of designing a translation start signal that'll give your protein to within a 4-fold range. So you can forward engineer with that predictability. This was summarized a number of years ago by a good colleague at Caltech, Francis Arnold, well-known in directed evolution research, with the following. There's no such thing as a standard biological part, because everything's going to behave differently depending on changing genetic context and operational context. So, key point. Let me just summarize and begin to wrap up. Although we can now construct, or more accurately, recapitulate genomes that are natural sequences, the forward engineering of even a single cistron is an open experiment. And that's significant because in the workflow of engineering, that means every time you want to go express one protein, you need to plan that you're testing whether or not that protein's being expressed. Now imagine trying to do that for an entire genome with 1 or 3 or 6,000 coding sequences. Very quickly, I'll return to this just to motivate how we get around these issues and how we can work together in getting around these issues. Here's the expression from those panels with the 2 different proteins shown differently. So just the scatter plot of GFP and YFP. If everything was well behaved, this would follow on the line y equals x. But on a log-log scale, you can see you get a 4 to 10-fold difference in the level of expression of GFP versus RFP for the same combination of control elements. We've tried to fix this. Let's just fix it. Let's see if we can. So one idea is we're just going to use the calculators and we're going to design what works, and we're going to design so many things that work. Many of them won't work, but we'll test them all, and hopefully we'll find something. So I'm going to skip over this one and go to what I just said. Here's the worst case scenario, in other words. You're never going to be able to make standard biological parts, but you use new technologies to make so many variants of things and test them all that you find the ones that work and then make a gigantic lookup table that virtualizes the idea of standard biological parts. So when a designer sitting at a computer needs something made, they'll have the one reagent in that context that works. To make this animated with an example, if you had a genome that you wanted to forward engineer that had 1,000 coding sequences, you would need to make 130 designs of translation start signals for each coding sequence, or 130,000 total, test them all, and some of those would work, and that would allow you to quantitatively tune protein synthesis across that entire genome. This is imaginable with some of the array technology coming out of Agilent, but it'd be a heck of a lot of work, so we haven't done it yet. Another idea would be to try and revitalize composition. So could we create a junction where the translation start signal wouldn't care about what came downstream? For example, could you bury the RBS in a structure that prevents any downstream structure from coming in when the coding sequence shows up? We tried this, it doesn't work. We don't know why. It'd be great collaboration with science to try and figure that out. We went to a different idea that's a very old idea. We went to this architecture. So if you want to translate a gene of interest, instead of just putting a start signal here, create a much more rich architecture. This is called a bicistronic architecture. You have a translation initiation signal here that doesn't initiate translation of your gene, but initiates translation of a throwaway cistron, only a few amino acids. Buried in this first leader cistron is a tunable second translation start signal, and you can vary this. The idea is translational loading here will linearize the mRNA and dominate any interactions that could arise between this RNA region and the variable region down here that changes. And the ribosome comes, it stops, it reloads based on the strength of this, and you get tunable expression down here. This is a very old idea. It's from circa 1982. It was never used to tune quantitative forward engineering translation, just for high expression. To make a long story short, we've tried this out, and here's what it looks like when you begin to make panels of proteins aiming for forward engineering of different specific levels. One protein over here mapped against 4 different proteins, and the scatter plot showing, without going into great detail, that we can get a correlation here greater than 0.93 across the panel. So we've changed how we think about putting parts together to create a more composable junction that simplifies the physics, basically, and the chemistry of how these things interact, and all of a sudden we can begin to do more forward engineering. Now, a 0.93 correlation is not good enough to get to genome scale engineering, right? It might get you to a pathway, and you build the whole pathway and test it all at once. Skip over this. Could we take this further? So the idea basically is to create a public architecture for laying things out on DNA. This is showing a single cistron or operon, but you could imagine scaling this to the genome scale and build it out and put it in the Commonwealth. Again, this could be done cooperatively. We've started work on this. So for example, here is out of 4,000 or 5,000 promoters we made, our top 300 that have different strengths and have different sequences at each strength level, so you could tune homology and direct repeats. We've done the same thing for transcription terminators within this context and so on, so not just translation CDS junctions. We begin to think about this as becoming the foundation of an expression operating system for genome scale engineering in the organisms you care about. Complexity. I've got maybe a minute or 0 minutes. So I'm going to skip over this. But, Dr. Post, rest assured we've been able to steal an idea from telegraph engineers in Britain in the 1800s and reduce context variation, which you often see in biology when you operate in different environments, from about a 10-fold level to about a factor 2. Maybe that'll come up later. The significance of being able to distribute measurements is we can coordinate work across place. And so I'm sure Dick will come back to this, but we've been able to do work in California and have that work be done in parallel in London and synchronize that, and that's quite significant. Skipping over this. The work that's been done that I've shown here is done by a public benefit factory. The people at this factory come from all over the world, and they're basically doing professional grade work to make these ideas come true. And I think that's an interesting idea. Like genome centers professionalize sequencing, we can have genetic engineering centers that professionalize synthetic biology. I'll skip over this. This work, as it gets started, is cheap. So the project we're running is $700,000 a year in cash from the public and $700,000 a year in contributions. And that's enough to begin to make an expression operating system. We have tremendous industry support, and it's essential to making this relevant. And I'll close then with this. If you think about innovation, innovation oftentimes is not us, them, private, public, open, closed, but most readily realized and most valuably realized in a rich ecosystem. And for example, here in computing, you see from the 70s, monolithic software development. In the mid-70s through mid-90s, bipolar, dysfunctional software development, competing models. And then today, a full translational ecosystem where you have private organizations making tremendous wealth, public benefit organizations fostering private wealth generation, but also many other people coming into the market. And I think we could do this with biotechnology if we organized our foundational work differently. Thank you very much.
Well, good morning again. It's a great pleasure for me to give this talk. What I want to do here is to return to some of what I think are some of the key conceptual points, and essentially to try and bring these out in my talk. So the structure of my talk is as follows: a bit of introduction, and quite a lot on systematic design, engineering biology, working with industry, the concept of national and international initiatives, and then just finishing up briefly with education and training. So first of all, introduction. Now, Paul has already shown this slide, but or at least in a slightly different form, but this is the definition which we work to within our centre at Imperial College, designing and making biological parts and systems that do not exist in the natural world using engineering principles, and it's the engineering principles that we are particularly interested in. So we see synthetic biology as effectively engineering biology. And what I would want to show in this next slide is what I think is the evolution of important ages in terms of science and technology, starting off with Huygens' pendulum clock in the middle of the 16th century, that you can think of as the analog age. Moving through into effectively the middle of the last century in terms of the digital age, which has resulted in modern telecoms and computers, etc. Norbert Wiener and Claude Shannon being a couple of the pioneers of that field. And then finally, moving into what I think we're moving into now, which is the biological age, as, if you like, evidenced by Watson and Crick's paper, and then the initial sequencing of the human genome. And it's really this area, if you like, which is the basis of synthetic biology coupled to the technology which is developing. Now, I think for me a very important point to get across is that I certainly believe that the endpoint of systems biology is the analysis of biological systems, and Paul has shown you the two sides of the same coin argument, but that the main endpoint of synthetic biology is industrialisation, and I can argue that within the questions. Another important point from my point of view is that systems biology is hypothesis-driven, whereas synthetic biology is applications-driven. Why is that important? It's important in terms of the way that the funders approach these two fields. It's very important, I think, to realise that synthetic biology is actually applications driven. Now, when we think about how we might develop a global infrastructure or a commonality in terms of approach for synthetic biology, what we see is a two-component strategy. The first component is the development of platform technology, so in other words, base technology. George was sort of touching on this actually earlier on, which goes across a series of fields, and then the application projects, particularly, and I'll come back to this, in collaboration with industry. So we think industry is very important in this. If you look at the evolution of synthetic biology, perhaps you can think about this as having a timescale of 30 years, say. We start off with genetic engineering, then going to metabolic engineering, and then finally, we would argue, moving through to synthetic biology. And I'm going to touch for a moment on the difference between these two and why it's significant. And this slide, this area has already been talked about, so I'll go through this quickly, but it's the area of malaria. And of course, the artemisinin, or artemisia in this slide story. Chinese herbalists, it says here, have known for 1,000 years actually that Artemisia, the annual wormwood plant, has an active ingredient which can be used against malaria. J. Kiesling and colleagues took this basic approach, as we've already seen, and produced artemisinin, and this is now in terms of is now being produced in terms of Amaris Biotech and a large pharma, leading through to a synthetic biology produced drug. But the key point here, which has already been touched on by both Paul and by George and Drew, is that it's taken 150 man-years to do this. This is actually a quotation directly from the paper in Nature from last year, Five Hard Truths, and something like $25–30 million. So how does the metabolic engineering and the synthetic biology difference fit in here? Well, we would argue that this fits in relation to systematic design. And the basis of much of engineering is this concept, and it is a concept of parts, devices and systems. One shouldn't take this too literally. The whole basis here is one of de-skilling, and this is the basis of the original Industrial Revolution, and we believe this is the basis of synthetic biology as well. So parts, devices and systems, for this audience it's almost not necessary to say this, but parts encoding biological functions, devices defining human functions, that should be sensors there, and then systems defining human-defined tasks like counting, controls, functions, etc. And at each level, and this is important, what we see here is that the characteristics of the part and the device are extremely well defined and reproducible. This is the engineering approach to how you tackle synthetic biology. A key element of this, we would argue, is the application of the so-called design cycle, starting off with specifications, leading through to design, a lot of computer modelling, implementation and testing, and one goes around this loop maybe 2 or 3 times. And what I'm showing here, and I'll come back to this, is actually one of our designs for a biologically based AND gate. And if we look over here at characteristics, I'll point this out now, but I'll come back to it, that this is not the characteristic that you want for an AND gate, although this is the biology working, if you like, in a natural way. So how do we do that? How do we carry out systematic design? Well, this diagram just shows this. Drew has already alluded to the concept of the registry of parts. What one does is to, in this approach, is to put biological parts together. I mean, that's not necessarily an easy thing to do, but it's not easy in other areas of engineering either. So if there are parts residing in the registry with the data and the metadata, they fit into the design. Otherwise, it is possible to actually characterize parts and then put them into the registry and use them in a particular design. So this is the basic approach to systematic design. Now this slide, which has come up at least once today, is a slide from the MIT Registry of Parts. And whilst the MIT Registry of Parts is very good for the iGEM competition, it has a number of issues related to it. For example, the data is often more qualitative than quantitative. It's been populated by iGEM students. Won't go through this whole slide, but basically, from using this from a professional point of view, we would argue is not the right approach. And what one needs to do professionally is to develop a professional registry of parts, which we, in conjunction with Drew and his colleagues and other people, are now beginning to do. So we're working in terms of this global infrastructure approach on developing a professional registry of parts which are fully characterized. And one of the key ways in which we can do this, and Paul showed this slide, we believe, is to use automation or to use robotics to do this. In other words, to take the human element out of this as far as possible. Now, why? Well, the reason is, and again, Drew has shown this slide, so this is a common characterization which we've done in London and in California. The reason for this is because what we want to do is to use the metadata here, which you certainly, I don't think, can read, but ultimately to program the robots actually in a similar way to the way in which MR scanners work, where you can program one MR scanner and you can program another across the Atlantic, for example, and get exactly the same results. The idea here is that with automation, the metadata and the characterization data, we can reproduce the same results in multiple locations. And that's the, if you like, the global approach that we're using. Another part of this is the development of web-based information systems. So we're commonly in a common drive, certainly with Berkeley and Stanford and Imperial, developing this so-called SynBIS system, information system, which is shown here. This allows you to have parts in a database, to view the parts, and also to compare the results of the parts, the way in which they work and the way in which the devices work, by comparing them to models, and then, if necessary, exporting those results. So this is work that's underway and is beginning to develop into something which is now working reasonably effectively. And this is leading to the concept of platform technology. So SynBIS is, if you like, the basis of storing the data and displaying it, but leading towards the concept of BioCAD in relation to DNA assembly, characterization, and then within the context of different chassis, how do these parts actually work? In terms of BioCAD, this slide is somewhat complicated, but it's just making the point that in terms of that there are really quite close similarities between the BioCAD approach and the approach which is used in terms of computer modeling, for example, in the systems biology area. And if you go across here, you can see where the similarities occur, but nevertheless, in terms of populating this space, there are already computer packages around, like Tinkercell, Cell Designer, etc., which populate these different areas, and one would expect that in the next few years that the rest of this will be populated so that you will have an equivalent facility as compared to the ability to carry out computer modelling, writing in high-level code, going down to assembly and machine code. That is certainly the objective. Here's an example of systematic design, just to bring out a key point. I'm not going to go through the whole exercise here, but this is the design of an AND gate. So this is a biologically based AND gate. Here's the truth table. What we actually did was to use this HRP regulatory system, and we separated out the two channels here so that we could get an AND gate type response. So you have two inputs. If you get the two inputs, 1 and 1, if you talk about it in terms of Boolean logic, then you get the output. So this is the basis of the AND gates. Now, what's key, and that's just an ideal AND gate. What's key here is that when we did the experimental work and we looked at different chassis, different sources of energy, if you like, and different RBSs, what we were able to do was to produce a series of characteristics. Now, the key point I want to get across here is here we have two different examples of the same design with effectively two different RBSs. In this case, RBSH in both cases, and then RBSH and RBS33. Everything else remains the same. From a biological point of view, you may think, well, this is not important, so what? But the key point here is that we're doing this from an engineering point of view, and now we get the characteristic that we want by getting these the balance between these two key RBSs. So the approach is different here in terms of how we engineer the biology to produce the AND gate. And similarly, we can produce an AND gate. So here are a series of characteristics which, from a biological point of view, are perfectly fine, but it turns out that one of them, actually one set of characteristics, works from an engineering point of view, being able to engineer the inverter we have here so that we can produce the NAND gate. And here's the characteristic. Okay, so working with industry, the main point of synthetic biology is industrialisation. This is the design cycle, assembly both small and large carried out, large assembly being carried out by companies leading through to industrial applications in terms of healthcare, pharma, biofuels, etc., which we are actively engaged in with a number of companies. Our strategy here is to develop a research pipeline leading through to an innovation pipeline, working with other researchers and then working with international industry in terms of innovation. Now, finally, one of the key points I really want to bring out, and this is a book called Crossing the Chasm, which is very well known in SME circles, there is a problem actually in terms of funding, which I want to bring out. So far, certainly let's say in the UK, that we have a significant amount of funding in terms of the basic research. But to move this to the next level, and I think Drew would probably support this, you need this combination of the university funding, industrial funding, and public funding. It's the only way that you can really get the catalyst working to de-risk the process. And my final slide here just makes the basic point that this is platform technology. This is going to work across a whole range of fields. This is not my slide. This is taken from analyst data, some of the Boston analysts that have looked at this. Thank you.
It seems to me that there is a particular responsibility for those of us who have been involved in the kind of engineering that has turned into synthetic biology to get it right, because there is an issue of public confidence in what we're collectively trying to do, and I think that up to now, as a community, we've been very careful, and rightly so, to make sure that what we do is transparent and assented to by people who are not necessarily scientists or specialists. And this is true of the idea of using synthetic biology or pathway engineering, or whatever you want to call it, to engineer the production of new drugs. And so what I'm going to talk about today has, in part, long roots in my personal research history. It, I hope, will tell you some new information, which I'm very excited about in this area. And I want to finish with what I think is a major challenge that we can now address because of the tools of synthetic biology. I don't say it's done, I just think it's possible. And I'm delighted I'm not the first in this meeting to put up Charles Darwin. Of course, we've been celebrating him, particularly in Cambridge in the last couple of years. And one of the first things that I want to remind you of is that natural products, that is the small molecules that you can consider as nature's chemistry set, are special because they are shaped and preserved by evolution. That is to say that they are genetically encoded and subject to all the pressures that every other constituent of life forms is subject to. And on the next page, I'm not showing you this for the iconic first picture ever of a phylogenetic tree, and I'm not going to ask you to read his scrawl here, But I'm showing you this because of that scribble in the margin, because he realized many, many years before he published, we don't have that luxury now, he realized that his theory implied that extinction had to happen. And so I want you to think about the evolution of natural products. I'm now converted to the idea that we shouldn't look for a billet for every bullet. We shouldn't look for a specific function for every natural product. Rather, it seems to me that Vernon Jones's idea that the trait that is evolvable and confers an advantage is the ability to produce such molecules. It's a sort of shadow, a foreshadowing, if you like, of the vertebrate immune system. We don't demand that every immunoglobulin has a specific role. And part of what I'm interested in is the natural evolution of these systems. But along with that goes the wish and the drive to engineer them ourselves. And those things go hand in hand. So I see every natural product as a representative of a virtual library of similar compounds that we could, because they're perfectly stable and sensible chemicals, could be recreated by us. And those of you who don't know what a polyketide is, you might at least see that it's a polymer, might well recognize the individual members of this large tribe of chemicals. We've already heard about rifamycin as one of the 4 components of a now 40, 50-year-old therapy against TB. Rapamycin is a soil bacterium of the Easter Island, produces this, and I'm going to come back to that. Epithyllone has featured already in a previous talk. Amphotericin, which is a ghastly drug that crystallizes out in your major organs, is still the drug of choice in UK hospitals for life-threatening fungal diseases. It's given in the form of liposomal preparations to try to make it slightly less unbearable. And in the middle is the molecule that I first got interested in. And if you know where to start at about 8 o'clock, you can follow the chain round. And if you strip off the sugars and you think about it, you can see that this is a polymer of one starter unit, basically a propionic acid, very, very simple acid, and 6 further 3-carbon units. So the number 6 is important because it's like a fatty acid biosynthetic enzyme. It knows how to count, and it does different things at each stage. And so let's just dwell for a moment on this historical finding. Much to our surprise, and at about the same time, Leonard Katz's group in Abbott Laboratories made exactly the same finding. It turned out that instead of using the same set of enzymes to build up a chain, which would have been very sensible and very economical, and to have the changing shape of the substrate itself dictate that different things happened. That was my original idea. What actually happens is that you use a different set of enzymes, very similar to the previous set, to add the next unit, and then you discard it. Or to put it another way, you actually move the growing chain through space along this track, exactly a molecular equivalent of a car assembly line. And what you see is what you get if you have more enzymes, and each of these circles represents an activity, then more chemistry happens. So you have full reduction here at module 4, whereas here you keep the keto group. If you kept all the keto groups, it would be a polyketide, and that's how they get their name. But actually what happens is that you get a mixture of things, a mixture of stereochemistries, and the number 6 is the number of modules, so of course you only add 6 and then it stops. And this immediately explained why hundreds, if not thousands, of similar complex chemicals in nature all are derived from a single simple pathway, by just modulating the detailed composition of the modules. So this looks like a type of enzymology where you have to redefine what it means to be a selective catalyst, because you're defined by where you are. and your substrate is presented to you. You're not given the universe of cellular components as potential substrates. You're given, in general, a single substrate. And what was immediately realized was that this meant that you could imagine tinkering with it. And so in the next few minutes, I'm going to talk about that promise of natural modularity. And you can see how this would fit with what we've talked about, and the problems with it. And then I'm going to encourage you by saying that the spin-out company, Biotica, has, despite those problems, managed to successfully create a focused library of bioactive polyketides that are going to be useful. And then I'm going to set a challenge. This is work that hasn't been done yet, but which I think synthetic biology makes possible. So here's the promise. This experiment was actually done. You rub out an enzyme that normally reduces a double bond. Everything else works, and you get an analog with a double bond which wasn't there before. And this is another very early experiment. Now that you've sequenced two different clusters, you can use one as spare parts that dictates a unit without a methyl branch. And when you substitute that in a little model synthase that makes a small ring compound instead of a macrocycle, a methyl group disappears, which, if you're trained as a chemist, as I was, has a certain charm, because a bacterium's doing it, and it can do it kilograms at a time in excellent yield. And that technology, called domain swapping, has been a mainstay of this field for 15 years. And indeed, people have thought and tried almost all the strategies you could possibly imagine, whether it's changing the nature of the extender units or the starter units or the oxidation states or the stereochemistry or the ring size, or indeed the sugars. I'm not going to talk too much about it. So it does work, and here's a real picture from a paper, not by us, with this sort of wallpaper design of about 50 new analogues. The snag is, if you read the small print, that they're not produced very efficiently. In fact, they're produced on alternate Thursdays in a mass spectrometer, if you're lucky. I rashly, to Leonard Katz, call this homeopathic quantities, and he said in his best New York accent, Peter says this is homeopathic, I call it homeopathetic. And he's right. So the reality is low yields. In fact, maybe wrong products, extra products, but very often nothing at all. And why aren't they modular? Well, it may be that the chemical that's attached, because remember the intermediates are not released, is simply not liked by the gateway ketosynthase of the next module, because this is the key enzyme. This is the one that's going to recognize the carrier and the growing chain on it, and is going to accept the elongating chain and do something to it. So if the ketosynthase says no, that's game over. And it could be that it doesn't like squares, it'll only have circles. Or it could be that the docking onto the acyl carrier protein is wrong because you've given it a different acyl carrier protein and it doesn't like it, even though it's a circle that's attached. Or it could be that the tether is not a blind tether, but actually an important piece of structure. And so, can we do better? Well, yes, we can do better in the ways that we heard about from previous speakers, that you can build up your databases and your methods of doing things. You can make synthetic genes, and people are doing that. But basically, we need structural information and a better knowledge base. And just to show that diagrammatically, there is now some, at least partial, X-ray crystal structure. Instead of a picture of each enzyme being loosely tethered in a very relaxed way, we now think there's very little wasted structure. So we can be more careful about the way we cut and paste, and that does mean we do better than we did before. And if you go down to the level of single amino acids, we know from work in Cambridge by my colleagues Kira Weissman and Bill Broadhurst that the reason that DEBS3 doesn't get out of turn and start docking on the end of DEBS1, and DEBS2 instead always docks on DEBS1, is because of an exquisite alpha helical handshake. in which, when you really look down at it, there is an electrostatic positive interaction when you have the right partner, and take my word for it, that isn't there when it's the wrong partner. And so what we learned in Cambridge and transferred to Biotica is this idea that this bit in here is quite tough. We can do some of it. But what we need to build up our number of molecules is to learn how to vary the front part, so the building blocks are different going in, and to build up the so-called post-polyketide synthase part, because then you start to multiply the numbers up. And here's Biotica's biography, and I don't want to take you through every word. We started it in 1996. We made some mistakes. We made a profit, actually. We paid taxes. That was a bad mistake, which we soon corrected. And then in 2000, we got a commercial contract from the business unit at Dow Agro. They were not sure whether to be pleased or startled when, after 18 months, we'd broken every one of their core patents on a molecule which for them was extremely important, a very nice compound called spinosad, which won the mother company, Dow Chemical, the President's Green Chemistry Award, which they'd never won before and probably won't again. And in 2002, we got funding, and I won't take you through all this, but let's just say our best, most forward candidates are now in advanced preclinical studies. So what are they doing? Well, the successful example that I'm going to quickly go through is rapamycins. Here it is, 14 cycles of polyketide chain extension and an amino acid. You don't need to know about the biology. It's perfectly sensible the protein target of rapamycin should be called the mammalian target of rapamycin. And it's absolutely key to the division of cells, for example, the cells of the immune system. So originally, rapamycin was an antifungal and then an immunosuppressant. This is a brutal summary of the fact that after several years, and not many millions, frankly, of money, they have 120 plus novel rapalogs in good yield by fermentation of the original host, which has been improved to meet the demand. And here's just one where you take away this methoxy group and you get something called BC210. You can feed to this so-called RAPK strain novel starter acids, and you do something called mutasynthesis, and you can feed the whole catalog if you want. Many go through, and just one piece of biological data. It simply says one BC210 partitions across the blood-brain barrier much more favorably than the parent molecule. And as an anti-cancer against a brain glioma, that would be good. And here's just a bit of fun. An experiment where nearly 2,000 mice were allowed to become middle-aged because they couldn't get the drug into the mice, and they discovered, nevertheless, they got a Nature paper out of it because the lifespan was increased for all these mice by an appreciable amount. And it hit papers all around the world, the Belgians in particular, elixir de jouvence, they had no doubt about it at all. And the exciting thing I want to give you 2 slides on is that antivirals are tough to find. And just recently, they have recycled a drug thrown away by Novartis 15 years ago because it was insoluble and it didn't fit the rules of 5 and all that sort of thing. And what this drug does, it binds to a host function that a virus needs, and that means the barrier for overcoming that drug is much harder. And they have good evidence that this compound called sangliferin, which was developed in combination with a lab in China, in Shanghai, the Chinese Academy of Science, and licensed to Biotica. And it's a totally different structure to the clinically tested cyclosporins. And we think it's got a lot of charm, and we've generated first and now second stage precursors. We think that in here there is a real opportunity for antiviral drug development. And this was really done on, I mean, we're not just lean and mean in Biotica, we're positively anorexic. And yet we can produce these things. So here is the challenge for the future. Even if you can tune and know about structure. An unsolved problem is that if a ketosynthase doesn't like what you're giving it, you will never have a really combinatorial production of new things. And so what we did was we tried to think if we could learn from nature. And while we were pondering that, some work that we did with Institut Pasteur scientists led by Stuart Cole and Caroline Demangel showed us that we actually had in our hands something that would fit the bill. Unfortunately, it had some problems. This is what happens when that polyketide is administered to a small human by the vector. We think it's a mosquito, so Luke, maybe we should talk. And borreliosa is actually more prevalent in West Africa than leprosy, and nearly as much as TB. And there's no drug therapy to speak of, no vaccine, you're disfigured for life. It's absolutely painless and there's no immune response. And the theory is that this is made by an assembly line enzyme too. This side chain as one assembly attached then to this core. And on the plane over to Paris, I wrote down the gene structure. And after a year, we saw the gene structure, and here it is. It's the biggest polyketide synthase and the most brutal that I ever had to try and sequence. And the reason is the internal homology makes it look as though it's evolved last week. There are 18,000 amino acids in this polypeptide chain. This is just a bacterium, and it's got 18,000 amino acids. And you can see I've highlighted the ketosynthases, and there are lots of them. All 16 are sequence identical, even though the substrates that they handle differ. And so it looks as though if you were to shut your eyes and change the position of any of those ketosynthases, it would still work. So maybe this is the combinatorial set that we've been looking for. But the trouble is, the biologists among you will know, if you put 16 sequences that are identical in tandem array, it will collapse. And so what we need is synthetic biology to synthesize these modules, varying the DNA sequence but keeping the protein the same. And that's what we intend to do. So that's a work in progress. And thank you for listening.
John McCarthy, University of Warwick again. With regard to registry of pre-built parts, at what point do you think, and this is a bit of thinking into the future, the usefulness of such part registries might be superseded by complete de novo synthesis from scratch?
It's a question that's a recurring question. Control of material can be control of a technology. Control of information, same, but is different and more challenging. So if gene synthesis today, or a few months ago, was $1 a base, let's say you wanted the top 1,000 parts from your collection, and each part has an average length of a kilobase, you're spending $1 million in aggregate obtaining that material. Let's say we project forward and the cost of building long-length fragments goes to a penny. Well, now the cost of that top 1,000 parts is $10,000 US, and so on. For some people, $10,000 is not very much money. For other people, it's an obscene amount of money. And so it's more a question of where are you on the financial spectrum, and how rich is the application you're trying to satisfy that begins to shape the answers to that questions. If you go to start a biosynthesis company in California for today and ask for money from an investor, the rules of thumb you encounter are the following: $1 billion market, 3 catalytic steps or less, and 90% profit margin. Right? So what that collectively does is disallow most biosynthesis research projects, because only a small number of them satisfy that requirement. Imagine doing software development, and it was $1 billion software market, 3 subroutines or less, 90% margin. You can easily get 90% margin, but you obviously can't do very much with software if you're only using 3 subroutines. So I think the answer to your question is, for some people it might go away soon. They'll be working on wealthy applications and can spend money. But for many other applications that are probably local, diverse, creative, esoteric, there's always going to be value in material collections.
And I mean, it's an evolving process, which I'm sure you'll aware of. I mean, I think for me, two areas which really give you a conceptual framework for sort of understanding how synthetic biology may develop are the semiconductor industry and also the aeronautical engineering industry. If you just take aeronautics as an example, I mean, going back to just the turn of the 20th century when the Wright brothers developed their first powered aircraft, we didn't stop producing aircraft because we didn't understand the physics of how flight works, or indeed how air flows across wings. I mean, there are hundreds of jumbo jets flying around even today, this very moment, and we still can't analyse the nonlinear equations that are associated with turbulence. So, there's this balance between progression in terms of what I'd loosely call an engineering approach and understanding the fundamentals, which is the balance between systems biology and synthetic biology in this field, in my opinion.
Let's have another question.
I'm talking from Shanghai. Today I can see there are two possible professional registries, and then is there any way they can be synchronized, which is a basic requirement for an engineering and for the most efficient usage of this registry?
Well, the answer I think to that is that these registries, I mean, Drew has alluded to the fact in his talk that some of this will need to be in a closed registry because that's certainly in the Western world, that's the nature of the way industry works. But I think many people in the synthetic biology field are working towards open registries, a lot of open information, and I believe those registries will then be potentially merged together. But if you're referring to, I'm going to express probably a somewhat controversial view here, but I am actually speaking as a professional engineer. I just don't believe that you can take the current iGEM registry and somehow morph that into a full professional registry of parts. Both those registry, the iGEM registry I think is excellent for the competition, but I personally think you've got to draw a line under that from the point of view of professional developments. And Tom Knight, when he gives talks, he often shows the Texas Instruments handbook from 20 years ago. And, I used to be a circuit designer at that time, and we never for one moment questioned that the Texas Instrument handbook was 100% correct. That's the level we've got to get to. Now, obviously we're talking about biology here, it's complex, etc. But fundamentally that's the context.
Hi, I'm Dr. Shiva Tambishetty from the London School of Economics. I'm just trying to tease out what the unpredictability might mean for, say, property rights. And the way I want to do this is to ask how unpredictability actually feeds into the peer review process when you publish papers in scientific journals. So is the unpredictability exciting in itself? Are you publishing things by saying, look, the margin of error is great, but by this paper we know so much more about unpredictability? I'm just trying to see if we can sort of measure it or quantify it, qualify it through peer review eyes on the progress that you're making. That's clear.
Yeah, that's a good question. We're shipping our work product out of Biofab Emeryville onto the web now. And one of the most important things we've advanced over the first 12 months is the idea that you could characterize the activity of a biological object not only on the basis of its primary biochemical or genetic activity, how strong is your promoter, but how much variance there is across local genetic context and operational context, strain, media, what have you. So if you think about in electronics, a resistor, you have oftentimes the fourth band, which is the tolerance, some measure of quality due to variation in the manufacturing process. We can now provide an analogous score for the operation of a genetic part, if you will. So it's an important idea. We'll publish the idea in a research journal, but the significance of the idea ultimately is that you can, right now, actually obtain parts that are unlike any other so-called standard biological parts. They give you not just an estimate of what they do, but how well they do that across context. To put that in the context of iGEM, I would agree with Dick. The iGEM registry is fantastic. It's hugely creative, but those students are not under a selection to evaluate the operation of their parts with every other part. And that's where we need to figure out how to establish professional research communities that can begin to try this stuff out, figure out where things break, and then, unlike in academia, not throw those breakpoints away because they're failures and you can't publish them, but collect those failures and disenchant them, if you will, debug them to figure out how to make those more reliable junctions. And I apologize for going through detailed work too quickly, but that's the lesson of what we figured out, that we could take a hairball junction and regularize it and improve its composition. We think you just need to do that again and again and again, and it's been a gap in genetic engineering research today. I don't know how this relates to property rights. All I can tell you is that for a couple megabucks a year, I can now foresee on a 12-month basis producing a self-consistent collection of control elements that take first-order engineering of the central dogma in your favorite organism much more off the table than it's ever been before. And that's not so much money that it's going to be impossible to get the money and give the stuff away, which is what we're doing.
Steve Latterman, Agilent Technologies. I have a question about the relationship of synthetic biology and systems biology. And so we've heard from numerous speakers this morning about very important and clear distinctions between synthetic biology and systems biology. And for sure, they're quite different in their motivations, in their methods, and in their results. And this is very important. At the same time, we've heard reference to some complementarity or perhaps some utility of systems biology tools and results in the context of at least some steps of the synthetic biology cycle. But since in total, systems biology oftentimes is really motivated by only a subset of the ultimate applications of synthetic biology, and in fact, a lot of it is focused on cells that are much more complex than the common chassis used in synthetic biology. And even some of the systems biology work that's on the more simple systems is in fact motivated.
There is a question
there
coming.
is motivated by a kind of using them as a test system to then migrate to more complex systems. I'm wondering if there's a gap or an opportunity for systems biology to be applied in areas that would be most useful for the purposes of synthetic biology that isn't really being filled or isn't filled as well as it could be for the synthetic biologists.
Well, I'll give you a quick example. So, I mean, certainly we have found within Imperial that by applying, synthetic biology approaches, that other colleagues in the biology department have sort of looked at this and said, oh yeah, well, we've got this work on different kinds of cells, which, has been kind of basically metaphorically parked in a cupboard for the last 10 years. And this looks really interesting in relation to an application area in synthetic biology. So I think there is a rich vein which one can mine here in terms of work that's already been done or work that needs to be done.
I don't mean to be crude, but I'll be short. Let me say it simply. Systems biology predates synthetic biology as a brand, and you can find examples of systems biologists commissioning or telling synthetic biologists what to do to advance systems biology. So make a more reliable genetically encoded oscillator, and do that by construction, and teach me about how systems work. Synthetic biology has not yet given marching orders of any significance at scale to systems biologists. And until that happens, I think there's an asymmetry in the partnership. That's not to say there isn't a lot there. I would like, for example, somebody in systems biology to advance a physics model for behavior of cells that isn't grounded in hard sphere dilute gas particle collisions, right? And apparent stochasticity from that physics basis, which doesn't make sense because it doesn't map to the inside of a cell. But we're missing that power right now in the relationship from synthetic biology back to systems.
To those one or two people, I did notice you out of the corner of my eye, but I'm absolutely positive if you hang on to your question, you'll get another chance in the next day and a half to ask it.